samples, more localities, more named taxa. These studies are finding evidence that not only is biodiversity underestimated, but it's kind of in many ways been misunderstood. So we have this fine scale phylogeographic structure in many cases, cryptic taxa in diversity. So we're kind of beginning to rethink diversity in this area. So I studied a group of birds called picards. These are suboceans in the genus Pachyranthus. Really cool group of birds, kind of taxonomically weird. The genus has bounced around a number of subocean families. But they're super widespread, that truly neotropical, ranging from Argentina all the way to northern Mexico, Texas. Within genus diversity is pretty complicated too. So you have relatively high diversity. 17 recognized species, a total of 55 named taxa altogether. So we're dealing with a large number of subspecies, and so it's a kind of a cool group to think about in terms of diversity. To give you an idea of some of this subspecies diversity, we're talking about some situations where you have kind of parapatric subspecies that are clinally variable in plumage, and in other cases you have these really geographically highly disjunct subspecies that are kind of phenotypically distinct and kind of cool too. So I tried to, the goal here was to try to characterize some of this diversity in Pachyranthus by looking for evolutionary taxa. And I characterize these by looking for things that are allopatric, so geographically separated, diagnosable, so things that are phenotypically distinct, and also reciprocally monophyletic. And so pretty similar to your standard phylo species, and these are hypotheses. So I'm looking for evidence of reproductive cohesion, evolutionary independence. And so I sequenced mostly mitochondrial DNA, 2 genes, ND2 and Site for about 265 samples, representing 51 to 55 named taxa. But I also sampled UCEs for 62 samples, representing 37 of the named taxa. And you guys have already heard a lot about UCEs. So I built a gene tree from the concatenated mitochondrial genes, also concatenated the UCEs. I built a tree in RAXML, and I also generated gene trees at each of the UC loci, and built a species tree in Astral. So, excuse me. So, to give you kind of a quick and dirty run through of the results, within the 51 named taxa or the 17 recognized species, I sampled kind of evidence for 40 evolutionary taxa. And if you kind of forget about the UCEs for a second, you just think about the mitochondrial DNA, and you do say like a Bayesian generalized mixed-field coalescent, you still find evidence of underestimated diversity, but it's turned out to be very conservative because it ends up lumping a number of recognized species. But what I want to do is I want to give you a few examples that I think are interesting from this group, examples where we've underestimated diversity. I can't show you the whole tree because there's a lot of diversity. But anyway. So this is Pachyranthus major down here, and Pachyranthus albobriceus. Pachyranthus major is found in northern Central America and Mexico. Pachyranthus albobriceus is found in Andes and in the Talamanca region of Costa Rica, western Panama. And in Pachyranthus albobriceus, you have a kind of deep, this is just mitochondrial gene tree here, but you actually find the same pattern in UCEs. So you find deep divergence across the Isthmus of Panama, and also subspecies that are kind of evolutionary independent, allopatric, and so on. What's also really cool about the species is it actually turns out to be paraphyletic or polyphyletic. So this species, or this subspecies guaiaculensis, turns out to be based on both UCEs and mitochondrial DNA based, turns out to be more closely related to the species group Pachyranthus polycopterus. And this subspecies is nearly identical phenotypically to salvinine. It's so phenotypically similar that we found specimens at multiple institutions in the collections that were misidentified guaiaculensis as salvinine. So we actually discovered a new kind of cryptic distribution that wasn't known before in this group. And here's another cool species, and I think this is a really complicated species here, where the mitochondrial DNA is actually a total mess, which is kind of cool, because you don't often see that. You have these parapatric subspecies in different colors here. Some of them are known to hybridize, kind of complicating the picture here. But we still find evidence of highly supported evolutionary taxa in the mitochondrial DNA that are also recovered in the UCEs up here as well. And so we have multiple taxa within these two species, row-sorted Picard and plain Picard. This is kind of the most complicated example, I think, in Pachyranthus. And what's kind of unfortunate here is I don't have the best UCE sampling for this part of the range, east of the Andes, which is what turns out to be the most complicated. And so, without kind of drawing too many conclusions, we have kind of a preliminary idea of the kinds of things that could be going on here. But west of the Andes, it's pretty clear. UCEs and mitochondrial DNA both show that these two subspecies are well supported. These are pretty distinct too. But this is a really complicated species. You have 8 subspecies altogether. East of the Andes, again, you have parapatry. Don't really know what's going on in part of this range. But within a single name taxon, a single subspecies, you have multiple evolutionary units. Again, this is mitochondrial DNA, so kind of take it as you will. And we need more data here. But it's just kind of cool to see these patterns. This actually tracks a known biogeographic pattern as well. So you have Western Amazonia, open area, and wet forest population, which is sister to Paraná population. And so that kind of supports it that you have independent taxa here as well. But again, I'd like to see this in a little more depth at some point. And if you assign tips in the species tree to the subspecies, you get kind of okay support in here based on subspecies names, but 93 and 84 based on over 1,000 loci is not really all that great. And so by making assumptions about the validity of some of these names, we may be misleading ourselves in the comparative analyses. And so I think the takeaways here, how am I doing on time? All right. The takeaways here are, first of all, there's lots of hidden diversity out there. A lot of it is going to be very young diversity. For example, in Pachyranthus, we have a preliminary time tree that puts the crowning age at about 14 million years. But a lot, even though that's kind of old for a genus, a lot of that diversity is really, really young. Some of it's highly range restricted. And so, in some ways, we need to start also rethinking kind of our ideas, rethinking some of our hypotheses about the generative mechanisms of this diversity as well. And I think the second point is, don't assume that the taxonomic limits are right because of long-standing names. And I don't think that that really comes as a surprise to a lot of people, a lot of systematists, but we're still using these names and treating them as real things and as real units of comparison. And that's potentially problematic. So the problem is, if we're getting the diversity wrong, or it's incomplete, we're getting phylogeography wrong, or it's incomplete, we're getting behavior wrong, or any comparative analysis wrong. And underestimation actually hinders our ability to conduct